Since the year 1953, when Watson and Crick discovered the double helical structure of the DNA molecule, scientists have been paving new ways to study the structure, sequence, and the function of this basis of all life forms. Therefore, since then, with a highly fast pace, new doors have opened in the fields of biotechnology and healthcare. If we look closer at the genetic information inside every cellular life form, at the top, we have deoxyribonucleic acid, or the DNA, that is made up of nucleotides containing 4 types of nitrogenous bases, namely adenine, thymine, cytosine, and guanine. These 4 components encode the information that provides the blueprint for the cell-direct physiology. Proper cell functioning requires proper DNA transcription and eventual translation into proteins. Any mutation or alteration in the DNA sequence in the genome could lead to possible cellular defects or diseases. Historically, the early attempts to determine the identity and correct order of bases present in a DNA fragment, referred to as first-generation sequencing, involved the cleavage of DNA fragments and using chemically altered nucleotides, followed by the manual separation of fragments generated via electrophoresis. These methods gradually evolved into a relatively applicable and somewhat scalable method called the Sanger sequencing. In this technique, using dideoxynucleotides or DDNTPs coated with radioactive or fluorescent molecules as highlighted in this picture, a single-stranded template DNA, and an oligonucleotide primer, the scientists were able to identify the sequence of the template DNA. The dideoxynucleotide can in fact stop DNA synthesis at their spot, the reason being that the dideoxynucleotide lacked the required 3' chemical structure that the DNA polymerase enzyme requires. Therefore, after the primer anneals to the template and the synthesis reaction is initiated by the DNA polymerase, the elongation of the new strand continues until one of these altered nucleotides enter the newly synthesized strand, which results in the termination of the synthesis process and the strand dissociates from the template. So, ultimately, using this technique, we will have thousands, if not millions, of DNA fragments with different lengths from the same template that have a specifically marked nucleotide at their 3' end. So, by putting all these tiny fragments together and sorting them according to their size, we will have the desired sequence of the initial DNA fragment. However, as revolutionary as this method was, the maximum read length using this technique was only about 1 kilobase pairs, which allows only for low-throughput sequencing. To sequence larger fragments or the entire genome, the DNA must be fragmented, cloned, individually sequenced, and then computationally assembled into genomes. Several global collaborations created methods such as automated Sanger sequencing. At the time, this approach was widely used to search for genes linked to human genetic anomalies. Later, next-generation sequencing, or NGS, also referred to as high-throughput sequencing, or second-generation sequencing, was developed. NGS uses a massively parallel sequencing approach with the ability to process large DNA samples at a significantly reduced cost and time. With NGS, millions or even billions of reads can be produced in a single run within a few hours or days, making it much more efficient than Sanger sequencing. To further put things into perspective, the sequencing of the human genome, which is about 3 billion base pairs long, using the 454 genome sequencer was completed in 2 months, with a cost of about $100,000. In contrast, it took 15 years with Sanger sequencing, required multiple collaborations across the world, and ended up costing approximately $1 billion. So far, several platforms of sequencers have been developed, each with their own advantages and shortcomings. However, they all can be put into one of the following 2 categories. Firstly, the short-read sequencing platforms, which include Illumina-based sequencers such as NextSeq and HiSeq sequencers, sequencers from Roche, and the Ion Torrent and Solid sequencers. The second category include the long-read sequencing platforms, for instance, Pacific Biosciences and Nanopore sequencing platforms. Now let's talk about applications of NGS in real life. But before that, make sure to subscribe to our channel and like the content if you find them helpful. In practice, to better understand the underlying pathophysiology of a disease with genetic background, we need to either study the sequence of genes or we have to assess the product of those genes in order to identify the disease etiology and/or its progression. Advances in sequencing technology did not just reduce the cost and streamline the process, they have also paved the way for new applications to elucidate molecular mechanisms of genomic structure and cellular function. So first, let's talk about the uses of DNA sequencing technology. Detecting DNA alterations that affect human health is now easily achievable because of NGS technologies. Sequencing of the entire genome or whole genome sequencing provides global information about coding and non-coding regions of the genome. Although comprehensive, the whole genome sequencing method can sometimes be costly and require extensive and time-consuming data analysis. For basic clinical applications, whole exome sequencing is a more cost-effective approach. In this method, only exons, which are the DNA sequences that are transcribed into mRNA, are analyzed. These sequences only make up about 2% of the human DNA. To further pinpoint the process, certain disorders already have commercially available targeted sequencing kits that cover a certain number of selected genes known to cause the said disorder. For instance, the otoscope targeted sequencing panels contain a list of genes known to cause hearing loss. This method has proven to be a very efficient tool in practice where time and cost effectiveness is of importance. The second major method in NGS is RNA sequencing. Early attempts to understand and quantify gene expression levels on the global scale relied on microarray technologies. One downside to microarray is that they require a sequence reference genome and transcriptome for its design and interpretation. NGS techniques such as RNA-seq can reveal the expression profile of organisms with an unsequenced genome. This method is more sensitive, provides better accuracy, and is not influenced by cheap sequence biases. This method can eliminate most of the background noises through high-throughput sequencing of cDNA fragments produced from RNA or RNA fragments, which results in an accurate mapping of transcript to unique regions of the genome. RNA-seq has been used for many applications in medicine, including the study of disease progression over time, such as heart failure. RNA-seq is commonly used to study differential gene expression, which can reveal expression anomalies or disrupted biological pathways. NGS technology has also been developed to study epigenetics, which are reversible changes to nucleosomes or nucleic acids that produce phenotypic variation without corresponding alteration to the genome. In general, eukaryotic DNA is organized into actively transcribed regions called euchromatins, which are less dense and more accessible regions of the genome. A technique to identify regions of euchromatin is assay for transposase accessible chromatin, or ATAC-seq for short. This method can identify and tag the open and active regions of the genome, and in the later process, these regions are then purified, amplified, and sequenced using NGS technologies. ATAC-seq can determine the location and interaction between transcription factors and shed light on how chromatin compaction is regulated. So finally, let's talk about how NGS can be used for the study of DNA-binding proteins. Another way to identify the regulatory process of the genome is through discovering the sequence of the regions of the genome where DNA-binding proteins are attached. And as for this purpose, chromatin immunoprecipitation sequencing, or ChIP-seq for short, is a commonly used method for capturing sequences with an attached protein. In short, in this technique, using antibodies, these proteins are first marked, they are then precipitated, purified, the proteins are then washed, and finally the remaining fragments of DNA are sequenced. As a result, the sequence of the regulatory profile of a given cell line will be identified. So as we saw, the next-generation sequencing is a relatively new approach, and though still a newborn, it has become one of the most important cornerstones in genetics and medicine. Wide applications for this method have been introduced, a few of which have been covered in this video. But stay tuned as we take deeper and more detailed look into how each of these techniques work and how we can use these methods in practice.